Hi everybody, I'm Florian, I'm here from Cambridge. I work for Cancer Research UK, it's a cancer institute. I will not be talking about cancer, so please don't tell my bosses. They will be very disappointed that I have side projects. So I was brought up a mathematician, but now I'm in a biological institute. I even have a wet lab. I've gone completely native. But when I think of biology, it still looks a bit like this. So it's colorful, it's cute, it's very complex, it's lively, and you always think something's looking out at you. What we try to do, people in computational biology and systems biology, is to kind of make sense of this complexity, try to understand how do the parts of the cell work together, how do these genes and proteins, just like we heard in the last talk, how do they work together to make cells live, to make cancers grow, to do all kinds of amazing and terrible things. Now there's different ways how you can approach that, different like philosophical decisions you can make. So let's ask somebody very smart. So Richard Feynman said, what I cannot create, I do not understand. So his idea is, well, you write down all kinds of equations, what mathematical biologists do, systems biologists, you have systems of maybe differential equations. You build up your system, and then you see what it does if you perturb it a little bit, give it different input values and all that. Now, interestingly enough, if you think of genetics and what we know about the function of genes, none of that was derived by building mice or building humans or building anything. What geneticists do is they break stuff. So if you cannot break it, if you cannot perturb it, if you cannot knock out a gene, if you do not have a mutant, you will never find out what a gene does. So what I will be talking about today is networks that are built from gene perturbation screens where you knock out genes and you look at the phenotypes of what's happening. And I won't be talking about mice. They're very cute, but they take so long to breed. They're quite it's very tough. What I'll be talking about is baker's yeast, which at least Wikipedia or Google or somebody tells me look like this. I don't think they look like this. Somebody colored them very artistically. So they are in everything from bread to wine, beer, everywhere. They're very easy to breed, and lots of stuff we know about, well, our human genes come from studies in yeast. So it's a very important organism. Now, key term today will be perturbations, and there's many different ways in how you can perturb these little critters. Some of it is like external perturbation, stress. You pour coffee on them, you heat them up, heat shock. Honestly, heat shock. That sends off a whole cascade of signals in the cell. What I'm mostly interested in today is knockouts of genes. So you take a piece of DNA which codes for a gene which is functional and important, you just put some random stuff in there. So the organism, after you did that, misses one of the different, in this case, 6,000 genes that it has, and that might have an effect or not. Now, after you did a perturbation, you need to observe a phenotype. You need to see what happened to the organism. And the simplest phenotype, what do you think is the simplest phenotype? 6,000 genes, not that many. You knock one out, what can happen? It's dead. So the simplest phenotype is called viability. Some genes might still be alive, for some others it will be dead. So if you only have 6,000 genes, how many do you think you can knock out and the organism survives? All of them? Nah. Well, one at a time, right? One at a time. If you knock out 6,000 out of 6,000 at the same time, okay. Now, you think half of them? Actually, it turns out when people did it, what you see is the fitness. Your fitness means you have populations of cells, you compare them to wild type, and you see after the knockout how many still survive. And so it goes from 0 to a bit beyond 1. And fitness of 0 means they're all dead, and this is about 1,000, a bit more than 1,000. So this is about 20% of cells. 20% of genes. If you knock them out, you kill the organism. The others are all over here. Fitness of pretty much 1. So you knock them out and nothing seems to happen, except for some of them here, which actually do much better than they used before. So this is amazing, right? So for 80% of the cells, it seems almost like the organism doesn't need them. So one interpretation is that this points off a lot of redundancy in the cellular networks of the cell. So just because you knocked one out, the cell still has a second way of how it can produce something important or how it can pass on some signal. So what do biologists do next? If they want to uncover this redundancy, well, instead of knocking out one, start knocking out 2 genes. So you start with your wild type, you knock out gene A and gene B, Yeasts are still pretty happy. And then you do the double knockout and see what happens. And sometimes, suddenly, after loss of 2 genes, the yeast dies, and that's called synthetic lethality. And in general, a genetic interaction is any kind of surprising result. And surprising here means that you wouldn't have expected it after you've seen the phenotypes of the single perturbations. Because you see, in this case, single perturbations say organism does well, and only when you do a double perturbation does it die. This is actually interesting for cancer research, because the idea is again that there's 2 different pathways that do some essential function, and only if you perturb both of them at the same time does the cell die. There's mutations in DNA damage repair in humans, in particular breast cancer. The BRCA gene is one of them, which impair DNA damage repair. But there's a second way of how cells can repair their DNA. Now, if you give a drug to cells that have a mutation in one of these DNA damage repair pathways and kill off the second way, then you kill the cell because it just accumulates so many mutations it can't deal with it. And that's called a genetic interaction. That's PARP inhibitors were very active field of research because they would selectively kill the cells that have this particular mutation. And that's different from chemotherapy, which is just really bad for you systematically. All right, so there's a little bit of relation to cancer here. Now, how do you do these kinds of, if you want to analyze a genetic interaction, what would you do? Well, you need different ingredients. You need individual perturbations, and you need a double perturbation. You need your wild type, and you need a phenotype that you can measure, and that can just be fitness, which is like how many cells survive. You observe the phenotype of the double perturbation, and given the 2 phenotypes of the single perturbation, you can compute some expected value, which very often biologists think in very simple terms. It's just adding up the phenotypes or multiplying them depending on whether you're on log scale or not. So this is often, if that's a reduction by 70% to 70% of cells, and that's 80% of cells, you multiply 0.7 with 0.8, and you predict the expected phenotype is 0.56, and then you compare it to what you observe. And the difference between expected and observed, that is the genetic interaction, which can be positive or negative. Okay. It's also called epistasis. Now, if you only have a pair of genes you're interested in, you still don't have a real big network, and you can do it by hand. What people are interested in is to do it for all the genes, for all pairs of genes in an organism, and that's where you need lots of robots and big labs. And you can turn individual epistatic experiments into a big network. There should be a little arrow here. So each edge in a genetic interaction network corresponds to one of these experiments I've just described to you. It corresponds to knocking out these 2 genes and comparing the double knockout to the individual phenotypes. And the weight of the edge would be something like the difference between the observed and the expected phenotype. All right. Shameless self-promotion. You can read all of that in this book. Now, people have done this. The nice people of the Donnelly Center in Toronto, they have dungeons full of robots and undergrads and helpless slaves who slave away to build all these big networks. And they published that a few years ago in Science. So this is a picture of let's get to that later, of a genetic interaction network in yeast. Every dot is a gene, and an edge indicates genetic interactions. They were also so kind to highlight some functional groups. So for example, all the yellow nodes here correspond to genes which have the same function. They're important for segregation and vesicle transport. And I don't know, the light green ones down here are responsible for DNA replication. So this is a picture I took from their paper. And for them, it's a very important picture. It's because it shows that this big network tells you something about the functional organization of the cell, because genes with similar function cluster together. So the way they interpret this, they say this network represents these different functions which are clustered together on it. Now, how do they quantify whether something's clustered on the network or not? Do they have p-values for that? Do we have any quantitative way of saying, well, for this particular function is cluster that is associated with my network, and this other function is not? They don't have that. They just do by eye informatics. They make this plot and they eyeball it. And that is okay. So clustering is a functional association. It's very important for biology. We want to understand these networks. What does the clustering depend on? Well, obviously, the network. Also, the subset of genes that we have highlighted. But, and that's the issue, the way they do it, also the visualization of the network. If we just plot it a different way, other nodes might lie next to each other. So we were thinking, can we come up with a statistical procedure, a test, if you will, that is dependent on the network and the gene set, but independent of the visualization? Okay. And what we want to know is this particular gene set very tightly clustered on my network, yes or no? Is that a question already? I'm not even finished. Yeah.
Sorry, so is it like weighted? I mean, what is it just yes or no, the edges, or are they? I mean, how do they? Are they just conveniently locating all the red ones in the same area? Is that what?
No. So.
What do you mean they're clustering?
So to do this, they had two inputs to do that. One is the network, which they experimentally derive. So the network is given.
Like a weighted network.
And the network in this case is weighted. Actually, I'll come back to that later too. So each edge is not actually an individual genetic interaction. It's the correlation between whole genetic interaction profiles. But it's a weighted network. And on top of that, independently of this, they have a whole list of gene sets with different functions. And then they pick one of their gene sets, and they put it on their network, and they color all the nodes. And in some cases, like all of the ones that you see here, you see that in this particular network, the genes that they have picked, they lie right next to each other. What they have not plotted, because it didn't fit into their story at that point of their science paper, is that there's many other gene sets which are just all over the place. So you would plot them, and there would be some down here and some over there, and they would not be clustered at all. So what we want to do is, if you have a network, and you give me one gene set, a subset of the nodes, I want to tell you, do I find evidence that is clustered, yes or no? Do they lie surprisingly close together? So how do we do that? So this is a method we call Santa. It had a reason, because we thought it would be out by Christmas, so 2 years ago or so, and it never was. Anyhow, so it takes as input a network, for example, the Costanzo network that's given. It also takes a collection of gene sets. We take them from the gene ontology, which is one of these biological collections. This is also given. We can look at it, but eyeballing is not enough. So we see, for example, well, red seems to be clustered, blue doesn't seem to be clustered. What I will describe on the next slides is a function called the knet function, which is has a big peak here because it's clustered, and it's very flat for the blue one. And if we do just resampling and we randomly throw subsets of equal size onto the network, we would find that the area under this curve is very far away from our random permutations, whereas the area under this curve is just in the middle of the distribution we randomly get. So this is the plan. So the missing bit now is, how do we compute this knet function here? Now, we got inspiration from spatial statistics, because clustering is something they look at too. They look at clustering on the plane. So they have dots here. And they want to know, are they just equally spread out? Are they randomly spread out? Or are they clustered together? And Brian Ripley has a statistic called the K-statistic, which looks like this thing down here. You go through all the points. You start drawing circles around the points, and you count how many other points do you gather in a circle of a given size. So you have a function that depends on the size, on the radius of the circles, and you walk through all the points, and you count how many other points are within this radius. If you plot this function against distance, you can use the area under the curve as an indication for how clustered things are, because if they lie closely together, then even for small radii, will you collect many points. Well, we thought we can do the same thing, we just do it on a graph. So we pick one node, we have a graph distance, you pick it, could just be shortest path, that's fine. And we just see how many genes in our gene set do we collect in our graph. And we have a pretty similar equation down here. It comes again in the next slide. We turned this by 45 degrees. We wanted it to be more like along the x-axis. So the ingredients of our function are this. this is the radius, which depends on the graph distance. You can use whatever you want. We generally use shortest path because it's convenient. We assume that there's weights at the nodes. That's like the phenotypes of our individual perturbations, and we want to collect them. If you just want to know if something is in a gene group or not, these weights would just be 0 and 1. We have some average phenotype and some normalization factors, and the inner product is interesting for other reasons because it tells you for each gene how central it is to all the other genes in this group. So if you had to prioritize a gene for your next experiment, the one with the highest inner sum would be the one you take. So here's 2 examples of why this is useful. So the genetic interaction network, I told you already, it actually shows the correlation between genetic interaction profiles and not directly the genetic interactions. The reason is that anecdotally people felt these correlation networks are more informative. So we thought, let's test that. So we derived 2 networks and chose the cutoff such that they had the same number of edges. One was the original genetic interaction network from the Science publication, and then the other one was the network of correlations. Just think of correlations between rows in the adjacency matrix. So the correlation matrix of these genetic interactions. And each dot here, okay, there's more dots on my screen than you see here. This looks really bad. This is not how it should look. But so the things you don't see, they're all up here. So they're all much closer. They're all above the main diagonal. And so we chose little bubbles because we wanted to show how big the gene sets are. We wanted to show we don't have a bias, that the big gene sets are treated differently from small gene sets and all that. But so that was important. So it means all the results, the p-values that we get from our resampling, they're all above the main diagonal, which indeed shows that the correlation network is more informative. Things are more clustered there. So the number of edges were the same in both networks. And then we compared 2 networks. One was a genetic interaction network in yeast, just normal yeast. And the other one was yeast again, but treated with a DNA damaging agent. And indeed, well, you can't see it, unfortunately, but stuff like response to stress, response to DNA damage stimulus, that were gene sets which were more associated with the treated network than they were associated with the untreated network. So we used that as some proof of concept that we can actually find interesting stuff. So there's lots of open questions with that. In spatial statistics, people think of boundary effects, and because you see you draw these circles and they can just go over the boundary of the area you look at, and they have to adjust for that. We don't do any of that in our graphs. Maybe we should. Edge density is an issue, of course, because you have a fully connected network and every node is connected to everything else. Everything is clustered. And if it's really sparse, then clustering will be much, much lower. So there must be a dependency of our statistic on how many edges you have in your network. There might be a sweet spot. When we compared the enrichment networks, we just chose cutoffs independently such that the number of edges was the same in both networks. There might be smarter ways to do that. And we never did any significance analysis for the difference. We just, again, eyeballed our scatter plots. And the validation, I mean, maybe it's a neat idea, but there's very few examples where you really have a biological network annotated in a way that you could say you know the true enrichment. So I'm just make up stories here, right? Okay. So that was my first story about Santa. Let's get back to this. So the phenotype we've looked at so far, the only phenotype we needed for all of this was survival of cells. But there are other phenotypes you can observe which should give you a much more higher resolution picture of what's happening in the cell after you perturb a gene. So for example, okay, this light yellow thing, which again you cannot really see, is supposed to be the cell. There's receptors in its membranes. That's how it senses its environments. There's pathways in the cell which relate this information down to the nucleus, and the nucleus have the genes which can be active or not. Now, if you perturb genes in different pathways, you will get different patterns of gene activity down in the nucleus. And that's not just one value telling you the thing is dead or not. This is like a whole long vector of values telling you for all the 6,000 genes whether they just went up or down. That's the gene expression profiles that Haiyan talked about earlier. Now that could be, for example, a readout, and that's much more informative, one would think, than just a binary dead or not decision. The problem is it can also be much more confusing. So people in Utrecht profiled yeast. They took 150 kinases, so that's proteins that work in signaling pathways. They did the individual knockouts, they did the double knockouts, and they looked at the gene expression profiles. For some examples, things were good. That's what they call complete redundancy. You knock out one gene, nothing happens. You knock out the second gene, nothing happens. You knock out both of them, boom, lots of genes vary. That's really cool. This is like synthetic lethality. Single phenotypes, pretty much non-observable, double phenotype big. Sometimes you see that, but it's more quantitative. The first gene doesn't do much. The second gene does a little bit more, but not as much as the double perturbation does. They are still happy with that. The problem is that most of the patterns they observed was what they a bit helplessly called mixed epistasis. It's just things are all over the place. So sometimes things go up, sometimes other genes go down. There's no observable pattern that they found easy to interpret. So we looked at this and we said, well, at least we have an hypothesis. We think we're not biologists, but we are good with making up stories. We think the fact that it's so complicated down here is because there's actually always a third gene which links between the two genes you have knocked out and the transcriptional response you see downstream. It's like a mediator. Question is, how can we identify these mediators? So what I will do is, I'll give you a short overview of a statistical method. That was my PhD, actually. It's called nested effects models, which is for inferring pathway structures from downstream effects, and then how we extended that to include some logics, and then use that to screen for modulators of genetic interactions. So a nested effect model, and talking to statisticians, I always feel a bit guilty because there's nested random effects models, and this has nothing to do with this, okay? I didn't know about the others when I named those. The idea is this. We have a network, we have a graph. That's the bold edges. That is the signaling pathway. That's what we want to infer. And we can perturb the individual nodes in here. But we cannot observe the effect one perturbation has on the other nodes in this network. We can only observe the effects it has downstream on transcription effects on reporters which are not part of the graph that we want to infer. So what we would like to have, what would like our life much easier, is like a little square matrix that tells me if I perturb A, then all the other genes in my pathway wiggle, and if I perturb C, it's only C and D that wiggle because they live in this branch. I don't have that. What I have is the effects of perturbing A, B, C, and D on a disjoint set of other genes of like downstream reporters, like gene expression, for example. Now, what my method does is it compares the expected data under a given model to the observed data, and if you have some idea on how big your false positive and false negative rate is, you can choose a model that reflects your data really well. So in nested effect models, the data are the states of the downstream reporters, which in the simplest solution are just binary yes, no, show the transcriptional change, yes or no values. The model is a directed graph. And what we model is subset relations, because when you look at it, if you perturb D, then you have effects at 5, 6, 7, and 8, which is a subset of the effects you see when you perturb A, which has effects at the whole range of reporters. Now, the way the reporters are attached to the graph is unknown. That's the parameters in my model, and the model itself is this directed, transitively closed graph. So the likelihood looks like this. There's one term which tells me for each effect reporter the false positive and negative rate, given that I have my model. I have replicate observations, hopefully, which I think are independent. I have different positions in my graph that I average over, and I have many of the effect reporters, and I think they're independent. In biology, they never are, but what can you do? Now, when you have that, in a way, you're done. You just need to search over the space of all graphs and find the best solution. That's the idea. And if graphs are small, it's not such a problem. Now, this brings us closer to what we want, because we have these perturbations and we have downstream effects, but we need to have kind of the double effects model, the double perturbations modeled. So in what I just showed you, if you perturb two genes at the same time, the effects would just add up. There is no logic involved. But if you had A and B, and in some logical sense they need to be combined to regulate D, then the effects at D, they're unknown until you specify the logic. And that could be an OR, an AND, an XOR. There are many different ways of what you could do. There are some logics which are more easily specified at just leaving out branches or edges in the graph. So when I talk about logics, I always talk about these 5. So what we did now is, So the only thing we actually did now is we took these graphs, we extended them by logics whenever one node had 2 parents. So we just expanded the model space a bit. And if we restrict ourselves to small graphs, that is just feasible to do exhaustive enumeration. And the likelihood function is pretty much the same because we still have an expected set of data and just compare it to the predicted one from our model. This is, love to blame it on the projector. There's supposed to be a network graph visible here. We selected some genes that some other people were interested in a Cell paper from some years ago, and we compared our results to all kinds of other results that were either published or done by other methods or that you could look up in the database. So for example, if you take the genes here, that's the pink ones, and you look them up in an interaction database, they just are completely connected. There's no structure and there are no interactions. If you just use a Boolean network on the data, it's not connected at all. That just seems to do something wrong. If you use a classical nest effect model where you do not have any of the logics, or pretty much all the logics are OR, it seems to fail. Lots of the effect sets are just very small in the end. So by looking at it, and again, without knowing the truth, we kind of felt our method was a better representation of the truth than all these other methods. So what do we do now with the genetic interactions? Well, if we have one genetic interactions and we have a transcriptional profile down here, we extend these 2 genes by a 3rd gene and the 5 logics that I have described, and we just search through all potential 3rd genes and try to explain the profile down here for one genetic interaction. And so, for example, when we do this for these 2 genes, so we have the log likelihood on the y-axis, and there's a certain set of potential modulators which show very high likelihood with different logics. Some are AND logics, some are masking logics like XOR logics, which explain the transcriptional profile very well. Some of the limitations are, well, the predictions were cheap to do. Took me an intern in 3 months. But actually following them up in the lab is quite hard, and we're working with people in Utrecht to actually do experiments based on that. But this is always the weak part, in particular if you're not in the yeast institute. I mean, if I go to my cancer colleagues and say, can you do a yeast experiment, they laugh, and that's the nice ones. The second problem is, so the logics, we used a very simple extension. We just have logics. If there's 2 parents in the network, you can, of course, the network could be much more complex, and we have no way of dealing with that right now. Good, that's it. Thank you very much for listening to me.
Questions? Perhaps I misunderstood, but I couldn't really follow what you thought that was wrong with the science paper, why you thought their analysis of the network was insufficient, and then what the Santa brought to it.
Well,
I mean, they have a distance matrix, and
there's nothing wrong with what they do.
I mean,
but they did it by eye, right? So
no, but that's not true. They're not just looking at the picture. That's just a representation. Obviously, they're doing some hierarchical clustering.
No.
Really?
I know them. I talk to them. So, I mean, obviously, they do all kinds of things. They do all kinds of things, right?
Why do you want to compare the data to the actual...
But this is, you see, there's no inference happening here. There's nothing, the data is all there. They interpret the data. Sorry, I should have...
But I mean, they do something similar to what you're doing, I suggest. I assume they're doing some sort of clustering of the data of the network, the weighted graphs, doing some clustering analysis on it, and seeing whether that compares to the clustering that's provided by the annotation.
Nope, definitely not.
So that is it.
That is it. That's the figure. They look at it and they say it's clustered. That's good.
So where do the colors come from?
Where do the distances come from then?
The distances come from. Okay, so
I mean,
you have a big square matrix. Every entry is like the epistasis from this genetic interaction, telling you the difference between your observed and predicted phenotype.
Sure. So how do they cluster on the basis of that?
And then, so this square matrix, now you do correlation coefficients between pairs of rows in there, And that is what each edge is here. That's a correlation between the genetic interaction profiles. And that gives you a network. You have a cutoff, so you don't, if they are too small, the correlations, you wipe them out. And then you just plot the network using a certain embedding of how to plot networks. They chose something that ends up in a circle.
Produce the image, sure, but I mean...
No, that's it. That's it. There's no other secret magic anywhere hidden there. They produce the image.
I would assume that they do some clustering, some hierarchical clustering.
On what?
Of the distance, of the correlation matrix, right? You plug that into some hierarchical cluster.
That would be a completely different approach. So, I mean, what you say makes a lot of sense. A lot of people, I feel, do that. So what lots of people do is they would look at this network and find little sub-networks and subgroups, and then later on take one of the subgroup they have inferred by clustering and compare it to a collection of functional annotations, which is the things in color.
Exactly, and you're telling me they did not do that?
They did not do that. So they colored it on the network, and they said, if it's clustered in my visualization, then there is an association between the I don't know, cell polarity, this gene set, and my network. And so what we did is, our contribution is, we said, well, we make it quantitative. I can now give you a p-value for this association. I have a quantitative measure of association where you guys had your eyes.
So no one's yet run any hierarchical clustering on that? That's my question.
No. I'm pretty sure tons of people have.
No one's done
No. Look, the genetic interaction networks, that was the one big thing yeast had in the last few years. That was the stuff you could get in a big journal. So the computational community have run every single algorithm you can think of on this.
Okay.
And now there's even a second paper where people quantify this clustering pattern. But this when this came out, the analysis was done by eye. No hierarchical clustering, no magic, nothing, no computation.
I haven't read the paper, but I find that hard to believe. I mean, it got into science for a reason.
Of course it got into science for a reason. Do you know how much work it is to make that network?
And they couldn't be bothered to do
I mean, come on, man. This is like years of work. You had to build up $10 million big robots. They have the whole basement of the Donnelly full of robots and undergrads running around. It took them 10 years to build the network. They wouldn't even have to plot the network to get into science. They were like, we are the first people who have a genetic interaction network within every pair of genes in a living organism. Science was like, that's cool, done.
I mean, one more question and then...
Yeah, sorry, I'm taking it.
I'm just curious, in your last part,
yeah,
you seem to be estimating large numbers of parameters, right? Didn't you worry about false positives and...
Yeah. For the you mean the nested effects when we have these modulators of...
the Boolean networks in the
yeah.
pool. The models.
So the number of parameters is actually... so the false positive and false negatives, we assume we know, mostly because we have control experiments. And we can see how many effects and differentially expressed genes turn up there. So these are not really parameters, they are a given. So the bad thing is the size of the model space. And in our case, it's pretty small, actually, because you have... two nodes are given, that's a genetic interaction. So you only have a single third one and these 5 logics. So this is the one thing we evaluate in one step. And that's true, then we do that for every potential third gene. So that's the big, but in general, these were not huge searches. So they were like 100 models or so we looked at.
All right, let's thank the speaker again.